Good afternoon, students. So welcome you all to another session. Right. So in the previous class, we started discussing about the disorders of the immune system. Right. So till now, what we have seen is that the immune system is responsible for providing resistance against any sort of diseases in our body. Sometimes what we have also said is there is this improper functioning of the immune system happen. So that improper functioning of the immune system would lead to some sort of severe diseased conditions in the human individual, which can even sometimes lead to the death of the person also. So those sort of disorders, we started calling them as immune disorders, or rather immune based disorders, which is mostly due to improper functioning of our immune system. So under that improper functioning of the immune system, we saw about three different categories. One, we called it as hypersensitivity or allergy, what we have already discussed in the previous session. The second thing what we have is called as autoimmunity. And the third one what we'll be discussing is with regards to something called as immunodeficiency, right? So in the previous session, we started discussing a few things with regards to hypersensitivity or allergy. So we saw what is this hypersensitivity all about. So we also saw how do we define hypersensitivity, and we saw what is the cause of this allergic reactions that occur in our body and what the immune system does to counteract with this allergens or any allergy causing agents inside our body. So that is what we have already discussed in the previous session, right? So today, let's take up with the second type of immune disorder that is there in our body, which is something related to autoimmunity, right? So let's begin with autoimmunity, which is the second type of disorder, which is actually seen. So what exactly is this autoimmunity all about? So whenever we talk about autoimmunity, things what you need to remember is this sort of immunity or this sort of disorder is usually caused due to some problems or some sort of defects during the process of maturation of the T lymphocytes as well as the B lymphocytes. Okay, let me make it very clear. Right. So what happens here in autoimmunity is, or rather we'll talk about the first natural process that occurs in the body, then we'll get into this. Normally, during the process of any sort of infection, what we have seen till now is the innate immunity will start functioning. Once the innate immunity is not able to fight against a particular pathogen, right? So we have the acquired immune system coming into picture. So the major cells of the acquired immune system are nothing but the lymphocytes. Right. So if you could recollect what we said is one of the characteristic features of the acquired immune system is to differentiate or to identify or recognize or to discriminate between the self cells and the non-self cells. Right. So whatever cells are present in the body, they are usually considered as the self cells. Whatever cells enter into the body, the foreign particles that enter into the body, they are usually considered as non-self cells. So that ability to differentiate between the self cells and non-self cell usually occurs during the maturation of the B lymphocytes and the T lymphocytes. Right? Let me come across it once again. Right? So the ability of the lymphocytes, that is the B lymphocytes and the T lymphocytes, to recognize, right, or to discriminate or to differentiate between the self cells and the non-self cells usually arises, or usually they start attaining that property during the process of maturation of the B lymphocytes and T lymphocytes. So we know B cells mature in the bone marrow, T cells they mature in the thymus. So during this process of maturation, they will attain the ability to recognize which is a self cell and which is the non-self cell. Right. So based on this, they will start fighting against the foreign particles that enter into the body. So during some time, right, during some sort of instances, during the process of this maturation of the T lymphocytes and the B lymphocytes, sometimes what happens is the T and B lymphocytes will start attaining certain receptors on their surface. Right, try to understand this. During the maturation of the T lymphocytes and B lymphocytes, right, during the process of maturation, some of the T and B lymphocytes, they would attain a receptor on their surface. So this receptor that is now present on the surface will now start recognizing the self cells as foreign cells. Getting it? Right? So let me come across once again. What exactly is happening is during the process of maturation, some of the T lymphocytes and B lymphocytes right? Some of the T lymphocytes and B lymphocytes, they would start attaining a receptor on their surface. Now, this receptor, say consider it to be a receptor X, right? So let us imagine that this is the T cell and this is the B cell, right? The T cell and the B cell. During the maturation of this T and B lymphocytes, say some of these T and B lymphocytes will start attaining, say, a receptor on their surface. Consider this to be the receptor, right? And this receptor on their surface, let us consider this to be receptor X, and this is also receptor X. Now, these two receptors will now start identifying our own body cells as foreign cells. Right, they will start identifying. So the T and B cells with these X receptors will start identifying our own body cells as foreign cells. So what would happen if they identify our own body cells as foreign cells? They would go and start targeting the self cells that is present in our body. But usually, most of the cases, this will not happen. Why this will not happen is because our immune system is created in such a way that, say suppose the T and B lymphocytes have this X receptors on their surface, majority of the cases, these T and B lymphocytes having these receptors would be suppressed, right? They would be suppressed. That is, their activity will not be shown. So this process of suppression of T and B lymphocytes with antigenic receptors on their surface, we start calling that process as clonal suppression, right? We call it as clonal suppression. So since the T and B cells are suppressed, what would they happen? They will not go and target the body cells. That's how our body cells are being protected from these particular cells. This is the first thing that happens to the cells, T and B cells, which have these sort of X receptors on their surface. Other case, what happens is, rather than bringing about suppression, these cells would be completely eliminated from the population. They would be completely eliminated. So this process of elimination of the T and B lymphocytes having these X receptors, we start calling it as clonal deletion. We call it as clonal deletion, right? So these T and B lymphocytes, if at all they have these receptors present on the surface, which would target the normal body cells, so those kind of T and B lymphocytes will be undergoing either clonal suppression or clonal deletion, so that they will no longer be able to target the immune cell. Sorry, they will no longer be able to target our cell. So this ability of our immune cells to identify and recognize the self cell without harming them, we start calling it as immunological tolerance. Immunological tolerance. We call it as, we use the term called as immunological tolerance. This is the normal thing that is happening. Right? So let me quickly go through it once again. One of the major characteristic features of our acquired immune system is to recognize the self cell and the non-self cell. So they have the ability to discriminate between the self cell and non-self cell. So that ability to discriminate between the self cells and non-self cell, we start calling it as immunological tolerance. Sometimes they attain this property of immunological tolerance usually during the process of maturation, right? During the process of maturation. Sometimes during the process of maturation, some of the T lymphocytes and B lymphocytes, they would start having certain receptors present on their surface. Now these receptors, that is here we denoted it as X receptors. So these X receptors would now start identifying the normal body cells as foreign cells, as a result of which they would go and target the body cell, normal body cell, and bring about the destruction of the normal body cells. But again, majority of the cases that process is not happening because the immune system will start suppressing those lymphocytes having these antigens through a process called as clonal suppression. Sometimes they are also eliminated from the population. We call that process as clonal deletion. So through the process of clonal suppression and clonal deletion, these lymphocytes having these sort of antigens will not be able to target our normal body cells. Therefore, the process of immunological tolerance is achieved. But again, during some extraordinary cases, right, during some very typical cases, what happens is these T lymphocytes and B lymphocytes, they would now escape this process of immunological suppression, and they would escape the process of immunological deletion, right? So they would escape the process of clonal suppression as well as clonal deletion. Now, what happens if they have not undergone clonal suppression and clonal deletion? What would happen? These T and B lymphocytes, since they have these receptors on their surface, they will go and start targeting the normal cells that is present in our body. So that process of immune cells targeting our own body cells, we call it as autoimmunity, self-immunity, right? So our own immune cells, they are now targeting our own body cells. So that process we start calling it as autoimmunity, right? So autoimmunity could lead to severe conditions in case of humans, which can even lead to the death of a person also at some point of time. So we'll see a few examples of this autoimmune disorders, right? So I hope all are now clear with what is this autoimmunity and autoimmune disorders all about, right? So normally the major role of our immune cells is to provide immunity or to provide resistance against a particular diseases without harming our own cell. But during some conditions, right? So during some conditions, what happens is they would start attaining certain receptors during the process of maturation, which would now recognize our own body cells as foreign cells. So for that, they have to escape the process of clonal suppression and the clonal deletion. Once they have escaped the process of clonal suppression and clonal deletion, then they would now go and target our normal cells, leading to a condition called as autoimmunity. Right? So this is what we have. The second type of immunological disorder or immune disorder, which is called as autoimmunity. Right? Now let's take up with a few examples with regards to autoimmunity. Right? So the first example what we have immunity is rheumatoid arthritis. Rheumatoid arthritis. Right? So this is one of the autoimmune diseases, rheumatoid arthritis, which is usually seen affecting the joints. Right? So what would happen here? The immune cells, that is the T lymphocytes and B lymphocytes, will start recognizing the cells in the joints as foreign cells, and they would go and target the foreign cells, bringing about the destruction of foreign cells, or destruction of the cells present in the joints. Therefore, we start calling it as rheumatoid arthritis, one of the autoimmune disease. This is actually considered to be the most common autoimmune diseases in most of the individuals. The second autoimmune disease, what we have, is called as multiple sclerosis. Multiple sclerosis. Right? So what is this multiple sclerosis all about? So multiple sclerosis is usually caused because of generation of antibodies, right? So antibodies are generated against the myelin sheath that is present in the neurons. So we know what is the role of myelin sheath that is present in the neurons, right? So myelin sheath is usually seen present in the neurons, right? So antibodies are directed against the myelin sheath that is present in the neurons. Therefore, they bring about complete destruction of myelin sheath. That condition, we start calling it as multiple sclerosis, right? So multiple sclerosis, antibodies are directed against what? Against the myelin sheath of the neurons. Multiple sclerosis. The third example, last example, what we can take is we have something called as myasthenia gravis. Myasthenia gravis. We have already come across this particular disease some time back, mostly in your first year, right? So we have a disease called as myasthenia gravis. So myasthenia gravis is also an autoimmune disease, right? It is also an autoimmune disorder, which is again caused because of the direction of antibodies. So here the antibodies are actually directed against, right? Antibodies are actually directed against something called as the acetylcholine receptors. So we know what is the role of acetylcholine. It is actually a neurotransmitter. It is responsible for transmission of impulse from one neuron to the other neuron. Chemical method, if you could recollect, we saw this previous year, right? So acetylcholine is a neurotransmitter. So for acetylcholine to bind with another neuron, there are certain receptors required. Only once they bind to the receptor, there is transmission of impulse happening from one neuron to the other neuron. So here, what happens in this disease, myasthenia gravis, is antibodies are directed against the receptors of the acetylcholine, against the acetylcholine receptors. Where is this acetylcholine receptors usually present? Mostly they are present in the muscular cells, right? In the skeletal muscles, right, present in the skeletal muscles. So once these antibodies go and target the receptors for acetylcholine in the skeletal muscles, that's what would happen. Skeletal muscles will not undergo the process of contraction. As a result of which, it would lead to paralysis, right? It would lead to paralysis of that particular muscle. Wherever these antibodies are being targeted, that entire part would be paralytic, right? It would undergo the process of paralysis because there is no impulse transmission between the skeletal muscle cell. So there is no process of contraction and relaxation of muscles happen. It would lead to a condition called paralysis, right? So 3 examples we can take up for autoimmune disorders. Along with this, we have a lot of diseases again, right? So we have something called as psoriasis is again one of the autoimmune disease. We have something called as uh, lupus, right? We call it as SLE, systemic lupus erythematosus, right? So that is again an autoimmune disease, right? So there are a lot of autoimmune diseases. Only thing what you need to remember here is our immune cells will start recognizing our own self cells as foreign particles, and they would start destroying our own body cells. That process we call it as autoimmunity. And the reason for autoimmunity is loss of immunological tolerance. Why there is loss of immunological tolerance? Because there is loss of clonal suppression and clonal deletion. So because of loss of clonal suppression and clonal deletion, the property of immunological tolerance is lost. Since the property of immunological tolerance is lost, what would happen? Our own immune cells, that is the lymphocytes, would go and start targeting our own body cells, leading to diseases such as rheumatoid arthritis, multiple sclerosis, myasthenia gravis. We have psoriasis, right? We have SLE, systemic lupus erythematosus, etc. Right? So this is what we have with the second type of disorder, immunological disorder, which is called as autoimmunity. Right? I hope all are clear with this concept, autoimmunity. Right? So let's quickly move on to the third type of disease or the third type of disorder, which is called as immunodeficiency. Right? So let's begin with the third type of immune disorder, which is called as immunodeficiency. So let's see what happens in this immunodeficiency. So from the name itself, we can make out deficiency. Deficiency is nothing but weakening, right? So there is weakening of the immune system. So why there is weakening of the immune system? It is because of the defects in either one component or more than one component of either innate immunity or acquired immunity, right? So due to defect in one or more components of either the innate immunity or the acquired immunity, we would get this sort of a disorder which is called as immunodeficiency. Right? Again, what is this immunodeficiency? It is caused due to defect in, right? It is caused due to defect in either one or more than one component of the innate immunity or the acquired immunity. It would lead to this sort of a disease called as immunodeficiency. Right? Now, this immunodeficiency could be broadly classified into 2 categories. Right? It would be classified into 2 categories. First one is called primary immunodeficiency. Right? So the first one is referred to as primary immunodeficiency. The second one is referred to as the secondary immunodeficiency. Right? So we have 2 types of immunodeficiencies. One is called primary immunodeficiency and the other one is called secondary immunodeficiency. Now, what do we mean by primary immunodeficiency? Right? What do we mean by this primary immunodeficiency? The immune deficiency or defects which are said to be either genetic or developmental defects. Developmental defects. Right? So here the defects in the immune system, that is either in one or more component of either innate immunity or acquired immunity, is seen because of genetic defects or due to developmental defects. Such sort of immunodeficiency, we start calling it as primary immunodeficiency. Right? So primary immunodeficiency is mostly caused due to either genetic defects or it is caused due to developmental defects, right? So the immunodeficiency that is usually caused due to genetic defects or due to developmental defects is usually referred to as the primary immunodeficiency. So what examples can we give you for primary immunodeficiency? We have one disease called as SCID. I hope you would have studied about this in your botany. SCID. So SCID stands for severe combined immunodeficiency, right? So which is again caused due to ADA deficiency gene, right? ADA gene deficiency in the ADA gene. It would lead to this disease called as severe combined immunodeficiency. So we are not going to get into much detail because hopefully you have already studied in your botany. So we are not going to get into the primary immunodeficiency. Now, what about the secondary immunodeficiency? Secondary immunodeficiency is actually caused, right? It is actually caused due to some exposure, right? It is caused due to exposure to what? Exposure to either chemical substances, right? It is caused due to exposure to either chemicals or biological agents, mostly the viruses, biological agents such as viruses, right? So the immunodeficiency that is caused whenever a person is being exposed, right? Whenever a person is being exposed to either chemicals or to some sort of biological agents such as viruses. That would lead to a disease called as secondary immunodeficiency, right? So primary immunodeficiency is majorly caused due to genetic or developmental defects, whereas secondary immunodeficiency is caused due to exposure to chemicals or the biological agents. So this is what we have with the secondary immunodeficiency. So what examples can we give here for secondary immunodeficiency? Examples what we have is AIDS. So AIDS fall under this particular category, right? So acquired immunodeficiency syndrome. So it falls under the category of secondary immunodeficiency. Along with that, we can also take the example of hepatitis. So hepatitis virus also falls under this particular category, that is secondary immunodeficiency, right? So this is the third category of disease what we have, right? After autoimmunity, right? So three types of diseases we said, immunodeficiency, immune disorders. Three types we have said. One was called hypersensitivity or autoimmunity. The second one here is what we saw is autoimmunity, and the third one here is said to be immunodeficiency. Under immunodeficiency, primary immunodeficiency as well as secondary immunodeficiency. Primary immunodeficiency caused due to genetic defects or due to developmental defects, whereas secondary immunodeficiency is usually caused due to exposure to either certain chemical agents or certain biological agents such as viruses. Examples what we have is AIDS. So we are not getting into much details about primary immunodeficiency because you have already studied it. What we'll do is let's get into the secondary immunodeficiency, right? So let's begin with this particular topic called as HIV/AIDS. So let's get into the details of HIV/AIDS, one of the secondary immunodeficiency disease. So under secondary immunodeficiency disease, we'll be studying about one disease in detail, that is nothing but HIV/AIDS. So let's get into AIDS. So I hope all are familiar with the abbreviation for AIDS. So this is the abbreviated form, AIDS. So AIDS stands for acquired immunodeficiency syndrome, right? It is referred to as acquired immunodeficiency syndrome. Now this particular disease called AIDS is caused due to exposure to a particular virus. So we said, right, it falls under the category of secondary immunodeficiency. Now when we mean that it falls under the category of secondary immunodeficiency, it is caused either to exposures to certain chemicals, number one, or it is caused due to exposure to certain biological agents. So here AIDS is actually caused due to exposure to a biological agent, a virus, which is called as human immunovirus, right? It is usually referred to as the HIV, right? It is usually referred to as human immunodeficiency virus. Right? Abbreviated as HIV, right? So this human immunodeficiency virus would lead to this particular disease called as AIDS, right? So this disease or this particular virus was first recognized in the year 1981, right? It was recognized in the year 1981 by an organization called as CDC, right? So CDC stands for Center for Disease Control, right? So this Center for Disease Control first recognized this virus in the year 1981. So let's get into the brief about this particular virus, and then we'll get into the AIDS part, right? So let's talk about this particular virus first, right? So we are talking about the HIV virus, human immunodeficiency virus. Now, this particular virus, HIV virus or human immunodeficiency virus, it belongs to a group of viruses called as retroviruses, right? It belongs to a group of viruses called as retroviruses. Now, what does this mean? What do we mean by a retrovirus? Retro means actually reverse. Retro means back. So we have studied a lot of things about retro, right? In our first year, if you could get back, we saw about something called as retrogression, retrogressive metamorphosis, all that we have studied. Retro means back. So what does this term actually mean? So if you could recollect what you have studied in your molecular biology, right? So you would have studied about the central dogma of molecular biology. So what is the central dogma of molecular biology? So we have DNA. All organisms usually have DNA as the genetic material, right? So this DNA would undergo the process of transcription, right? So this DNA would undergo the process of transcription to form RNA, more precisely speaking, the mRNA, the messenger RNA. This messenger RNA will now undergo the process of translation, right? It would undergo the process of translation and form the specific proteins, right? It would start forming proteins, and these proteins could be of two types. You would have studied about this, right? They are of two types. One is called the structural proteins, other one is called the functional proteins, all that, right? So this we start calling it as the central dogma of molecular biology. DNA, through a process of transcription, would form mRNA. mRNA would now undergo the process of translation to form the proteins. So this is the normal mechanism of protein synthesis in almost all the organisms present on the planet Earth. Right. So majority of the organisms, they have DNA as the genetic material. So that DNA would be first transcribed into mRNA, and that mRNA would undergo the process of translation to form the proteins. Clear? But in case of this virus, it is not the same thing. They don't follow this central dogma. It is quite different. In what way it is different? The genetic material present in this particular organism, HIV virus, is not DNA. Rather, it is RNA. Right. So the genetic material here, in case of HIV virus, is RNA. So RNA is the genetic material. Now, if RNA is the genetic material, what they should do first is this RNA would first be transcribed, right? It would be transcribed into DNA, right? So RNA is first transcribed into DNA. This process here, DNA to RNA, we call it as transcription. But here, the RNA to DNA, we would start calling it as a process called as reverse transcription. Right? So they would undergo the process of reverse transcription. So through reverse transcription process, the DNA is being converted to RNA. Now this RNA is being converted to DNA. Now this DNA would now start undergoing another round of normal transcription to form two types of RNA. One would be called the messenger RNA, mRNA. The other one would be called the genomic RNA. So it would form messenger RNA and the genomic RNA. This messenger RNA would now undergo the process of translation to form the proteins. So this is how they usually start undergoing the process of transcription and translation. Along with the normal process of transcription and translation, they have one additional process which is called as reverse transcription. Normally, it is from DNA to RNA, but here what is happening? They are moving from RNA to DNA. So they are moving in a reverse direction. They are moving in a retro direction. That's the reason why they had called it as retroviruses. Right. So retroviruses is usually seen when an organism has RNA as the genetic material. Right. Since RNA is the genetic material, RNA should be first reverse transcribed to DNA, and then the normal process usually occurs. So since in this organism, RNA is the genetic material, and they undergo the process of reverse transcription to form the DNA, they are usually referred to as retroviruses. Right? So this HIV virus, it belongs to a group of viruses called as retroviruses, where RNA is the genetic material. Right? So this is what we have a brief introduction with regards to this particular virus called as HIV virus. Now let's get into the structure of HIV virus. Right? So let's get into the structure of HIV virus. So what does this HIV virus consist of? So here in AIDS, we are talking about the HIV structure. So let's get into the structure of HIV virus. So what does this HIV structure or HIV virus structure include? So let's move from outside to inside. So what is present in the outside? Then we'll move on to the inside. The outermost layer, one more thing. This particular virus belongs to a category called as enveloped virus. So what do we mean by enveloped virus? All viruses having an envelope, a bubble-like structure around them. We start calling them as enveloped virus. So this virus also belongs to the category of envelope virus. So this is the envelope, right? So this is the envelope of this particular virus, right? This is the envelope, right? And this envelope is a double layer, right? It is actually a double layer, and it is composed of phospholipid bilayer, right? So this would be the envelope, right? Envelope, right? And it is nothing but a bilayer, right? It is a phospholipid bilayer. Now, from where do the virus get this envelope or this particular phospholipid bilayer? It is actually considered as a stolen material. Stolen material of what? Stolen material of the plasma membrane of the host cell. Right. So if we see, once the viral particle enters into the body, it definitely enters into some cells that are present in the body. So which cells does it enter? It usually enters into majorly cells having CD4 receptors. We'll talk more about this once we get into the life cycle. For now, remember that it first enters into cells having CD4 receptors on their surface. One of the cells having CD4 receptors we have already discussed, T helper cells, right? So T helper cells have CD4 receptors, right? So the outer envelope of this particular virus is nothing but the plasma membrane of the plasma membrane of the T helper cells. So it is nothing but the phospholipid bilayer or the plasma membrane of the T helper cell, which is the envelope. Now, on this envelope, there are two types of proteins which are being present. We call them as glycoproteins. Right? There are two smaller units of glycoproteins which are being present. So here there is one unit of glycoprotein present. So this is the first unit of glycoprotein, which we usually call it as the transmembrane protein. Why do we call it as transmembrane protein? Because it is present on the phospholipid bilayer. We start calling it as transmembrane protein, and this protein is usually referred to as GP41. So GP stands for glycoprotein 41. We will be seeing different numbers here. So all the numbers are usually given based on the molecular weight of that particular component. So this is one protein which is said to be a transmembrane protein originating from inside, and it is seen on the plasma membrane lipid bilayer, and it starts projecting to the outside. It is called as GP41, glycoprotein 41. Right. Now, covering this glycoprotein 41, there is another protein that is being present. Right. So there is another protein that is being present here. So this is the this is another glycoprotein that is usually seen on top of this GP41. Right. So this protein here is referred to as GP120. It is called as glycoprotein 120. Right. Most of the cases, these two proteins together, we call it as GP160. One single unit, we start calling it as GP160. It is a large polypeptide, and this large polypeptide is broken down into two smaller peptides, two smaller proteins, GP120, which is present towards the outer side, and GP41, which is present towards the inner side, a transmembrane protein. Right. So GP120, one important thing what to be noted down here is GP120 is the one which usually attaches to the CD4 receptor on the host cell. Right. So it is also called as attachment protein because GP120 is the one which attaches itself to the CD4 receptor. If this is CD4 receptor present on the T helper cell, right? So we have this is the GP120. It goes and attaches to the CD4 receptors present on the immune cells, either the T helper cells or the macrophages or that. So these are the two proteins which are present on the plasma membrane towards the outer side of the body. Now let's get to the inner side. towards the inner side, just attached to this GP41, right? Attached to the GP41, there are certain proteins which are actually being present, right? So there is this protein which is being present. So this protein that is present towards the inner side, right? So this protein that is present towards the inner side, we start calling it as matrix protein, right? It is called the matrix protein, right? And it is also referred to as P17, protein 17, right? Matrix protein. So this would be the matrix protein, which is called as P17. Again, as I said, 17 is nothing but the molecular weight of that particular protein, right? P17. Now, what is the role of this P17 protein? The major function of the P17 protein is to stabilize GP41, the transmembrane protein, right? So this P17 usually stabilizes the transmembrane protein called as GP41. So three proteins we have seen so far. GP120 towards the outermost side, we call it as attachment protein. Then we have one more, GP41 present towards the inner side and the outer side, we call it as transmembrane protein. Then we have protein 17 or P17, which we call it as the matrix protein. And this matrix protein is responsible for stabilizing the P41. Right, so this is what we have. Now, towards the inner side, the actual virus particle is actually present towards the inner side. Now, what is present? Towards the inner side, right, towards the inner side, there is this house-like structure that is present. Inside this house-like structure, we have all things that are actually being present, right? So, we have a lot of things actually present inside this house-like structure. So, we'll see what is this and what does this consist of. This structure, we start calling it as the capsid protein. We call it as capsid protein. And this capsid protein is, again, since it is a protein, it is made up of something called as P24, right? Capsid protein is nothing but P24. So here we have P24, 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 and P24. All these are nothing but capsid proteins, right? So P24, we call it as P24 proteins, or simply we can call it as capsid proteins, right? Now this capsid protein, entire capsid structure, if we see, it is a 20-sided structure. Just like how we have triangle, 3-sided structure, square has 4 sides, like that. This structure, capsid, is a 20-faced or 20-sided structure. And that 20-sided structure, we usually call it as icosahedral, right? So icosahedral, right? Capsid protein structure, if we see, it is said to be icosahedral because it is a 20-sided structure, right? Icosahedral. So if we see under a microscope, electron microscope, it appears to take the form of a cone. It is actually a cone-shaped structure, but actually speaking, it has 20 sides, the capsid protein. Now, inside this capsid protein, the actual substances are present. Now, what all are present inside the capsid? Inside the capsid, there are two molecules of, right? There are two molecules of single-stranded RNA, right? So there are two molecules or two copies of identical single-stranded RNA molecules, right? So inside the capsid, right? Inside the capsid, there are two molecules of single-stranded RNA molecules that are being present. Right? Again, the important thing what you need to remember, most of the other viruses, if we see, if there are two copies, they would be complementary. But here, they are said to be two identical copies of RNA molecules are present. Right. Now, along with these two identical copies of RNA molecules, what else is present inside is they have three important enzymes being present. Right. So there are three important enzymes. Very easy way to remember these three enzymes. Number one, the first enzyme is said to be R. It stands for R. Right. So R stands for reverse transcriptase. Right. The first enzyme is reverse transcriptase. So this is the first enzyme that is actually present. The second enzyme that is present stands for I. right? It stands for I. So what does this I stand for? I stands for integrase, right? Reverse transcriptase, second enzyme is integrase, and the last enzyme is P. So you can just remember it this way, RIP, right? So P stands for protease, right? So three enzymes present inside the capsid. One is called reverse transcriptase, second one is called integrase, and the third one is called protease, right? So what is the role of reverse transcriptase? We have already seen what does the reverse transcriptase do. Reverse transcriptase is responsible for bringing about the process of reverse transcription, where RNA is being converted into DNA. Second one, I stands for integrase. So what does integrase mean? From the name itself, we can make out it brings about the process called as integration. Integration of what? Integration of viral DNA into the host DNA. We'll see what exactly is happening once we get into the life cycle part. The third one is protease. P stands for protease. What is the role of protease? In general, we know that protease is responsible for the breakdown of polypeptide. Right, large protein chain is being broken down with the help of the enzyme called protease. So that is the role of protease. So what all are a major constituent of this viral particle? Two single-stranded identical copies of RNA molecules, then three major enzymes: reverse transcriptase, protease, and integrase. So once we get into the life cycle, you'll get a clear picture of what is the role of each of these three proteins, right? So in general, in brief, this is what we have with the structure of the HIV viral particle. So what all does the HIV viral particle consist of? Basically, it consists of an outer structure called as the envelope. So that envelope is things what you need to remember. It is actually a stolen material. Stolen material of what? Stolen material of the host plasma membrane. Right? Stolen material of host plasma membrane. This envelope consists of two smaller units of proteins. One is called GP120 and GP41. GP120 is responsible for bringing about attachment to the CD4 receptors present on the host cell. GP41 we call it as transmembrane protein, which is actually present on the protein envelope, right? On the envelope. So these two together we call it as a larger unit, which is called GP160. Right. Moving to the inner, we have one protein called as the matrix protein, right? Matrix protein, which is also called as P17 protein, right? So this matrix protein is responsible for stabilizing GP41, which is the transmembrane protein. Inner to the matrix protein, there is another protein which is called as capsid protein, which we call it as P24 protein. Clinically, there is a lot of significance for this P24 protein, right? Especially during the process of diagnosis, right? Diagnosis, we have two important methods to diagnose HIV. One is called ELISA, which you would have already studied, and the other one is called Western blot. I hope this also you would have studied. Two important ways how we diagnose this particular disease: through ELISA, which we call it as preliminary confirmation, and the last one is the confirmatory test, which is the Western blot. So in both the cases, antibodies against P24 is the one which is identified to detect that particular disease. So clinically, there is a lot of importance for P24 protein. So inside this capsid, that is P24, one more important thing with regards to this P24 protein, structure is said to be icosahedral, right? Because it is a 20-sided structure, right? Inside this capsid protein, right? Inside this capsid protein, there are the major constituents of this particular virus. So what all are present? There are two copies of single-stranded RNA molecules that are present inside, and along with that, there are three important proteins, which we usually call it as the RIP proteins, RIP proteins. So R stands for reverse transcriptase, right? I stands for integrase, right? I stands for integrase, and P stands for protease. Reverse transcriptase bringing about the conversion of RNA to DNA. This RNA will be converted to DNA with the help of reverse transcriptase. Integrase brings about the DNA integration with the host DNA, and P stands for protease, which is responsible for the breakdown of larger polypeptide into smaller proteins. Right. So this is what, in brief, the basic structure of the HIV particle what we have in simple. Right. So in the next session, we'll take up with the transmission of this particular virus. So how is this virus being transmitted from one person to the other person? And then we'll take up with the life cycle of this particular virus. So how exactly or what happens to this virus once it enters inside the body, right? Then we'll also see how to prevent this disease and how to what is the treatment options that are available and how do we diagnose it and what are the symptoms of all of this particular disease, right? So with this, we are done with the structure of the HIV particle, right? So in the next session, let us take up with the life cycle of the HIV virus.